Chapter number 3. Tools of Genetic Engineering. Basic requirements. Genetic engineering equals gene cloning. Our DNA technology can as changing of genes by using in vitro processes. A gene of known function can be transferred from its normal location into a cell that of course does not contain it via a suitable vector. The transferred gene replicates normally and is handed over to the next progeny. On confirmation for its presence through biochemical procedures, replica of the same cell, i.e. clones, can be produced. The derivation of procedures for the reintroduction of the foreign DNA fragment into a bacterium have led to evolution of new technology, i.e. the recombinant DNA. Technology, gene cloning, gene manipulation or genetic engineering. The macromolecules such as DNA, RNA, proteins, etc. are synthesized inside the living cells which vary with each other in respect of molecular weight, size, solubility, presence of charges, absorbance of light wavelength spectrum, etc. There are many techniques that are used to isolate and characterize the macromolecules on the basis of differentiating features. Separation and characterization of macromolecules are discussed in this chapter. Size of different types of molecules varies and, therefore, their molecular weight also varies. For example, a macromolecule of small size shows less molecular weight and vice versa. Techniques used on the basis of molecular weight are gel permeation, osmotic pressure, polarity of charges. A gel permeation or gel filtration. In this technique, polymeric organic compound is used to prepare a porous medium. The polymers form a three-dimensional network of pores. The pore size is determined by degree of cross-linking of polymeric chains. Solutes present in the mixture are separated on the basis of their size and shape when they pass through a column consisting of packed gel particles. Figure 3. For such separation, many terms like exclusion chromatography, gel filtration, and molecular sieve chromatography are used. The small-sized molecules enter the gel beads, whereas the large-sized molecules remain outside. Figure 3.1. Solvent, usually a buffer, is used as an eluent. The small-sized molecules are distributed between the mobile phase, liquid, inside and outside of gel particles. V0 refers to the volume of the mobile phase outside the gel particles, where V is the volume of mobile phase present inside the gel particles. Different types of molecules in gel are separated from each other. Distribution coefficient Kd of molecules for a given gel depends upon their size. Very large molecules have no access to mobile phase within the gel, hence their Kd value is L. The small molecules have accessible inner mobile phase. Therefore, their Kd is 1. The Kd values of molecules govern their separation. Separation of macromolecules by using gel permeation offers many advantages such as 1. Separation of labile molecules, 2. Recovery of solutes in maximum quantity, 3. Short time and expensive equipment, and 4. High reproducibility. Characteristics desired for gel filtration media. There are certain characteristics desired for gel filtration media such as 1. Inertness of gel matrix, 2. Presence of low amount of inorganic groups, 3. Uniform pore size, 4. Wide choice of gel particles and pore sizes, and high mechanical rigidity. These criteria can be fulfilled to varying degree by some of the media such as Softex. Application of gel permeation. Gel filtration is used for several purposes as given below. Desalting or group separation. In many experiments, e.g. protein separation, inorganic salts are used. Salts should be removed during final preparation by passing through a column of Softex G. 10. Fractionation or purification. Viral particles and low molecular weight compounds like sugars, proteins and nucleic acids of varying molecular weight can be separated. Determination of molecular weight. The macromolecules are separated on the basis of their relative size. If molecular weight of a compound is large, it will have lesser elution volume. This method has been used to determine the molecular weight of protein from their elution characteristics via Kd value. Ion exchange chromatography. When molecules are dissolved in solvent, they dissociate into charged ions. Therefore, they develop polarity. On the basis of polarity, i.e. presence of charges, they can be separated. Differences are found in charges, charge densities and distribution of charges on the surface of different molecules. These differences in charge properties of different molecules are taken into account to separate from the other identical molecules. Ion exchange chromatography is a powerful technique used for separation of two proteins which are very similar to each other, but different from each other in having one charged amino acid. In ion exchange chromatography, solutes are retained on an ion exchanger due to their reversible interaction with the oppositively charged groups on ion exchanger. Ion exchangers are prepared using some synthetic resins which must be insoluble porous organic molecules. The ion exchanger consists of synthetic or normally occurring organic biopolymers as insoluble matrix to which various groups known as fixed ions are covalently attached. These fixed ions are balanced by equal and oppositely charged mobile ions from the solution called counter ions. On the basis of counter ions, the ion exchanger has been grouped into two types, the cation exchangers and anion exchangers. Figure 3.2. Ion exchange chromatography. A cation exchanger, anion exchanger. A. Cation exchangers. These are negatively charged exchangers containing positively charged counter ions, cations. Figure 3.2a. On the basis of strength of charged functional groups, cation exchangers are divided into three types: strongly acidic, intermediate acidic, and weakly acidic cation exchangers. B. Anionic exchangers. These are positively charged exchangers containing negatively charged counter ions, anions. Figure 3.2b. On the basis of strength of charged functional groups, anions exchangers are also divided into three types: strongly basic, intermediate basic, and weakly basic anion exchangers. C. Matrix quality. Inorganic compounds, synthetic resin or polysaccharide based matrix are used in ion exchangers. The chromatographic properties, e.g., efficiency, mechanical strength, chemical stability, flow properties, etc., of exchangers are governed by the matrix. The matrix also influences the biological activity of molecules. The basic three groups of materials used in construction of matrix for ion exchangers are cellulose, resins, polystyrene or polyphenolic, and polymers or dextran or acrylamide. Table 3. L. D. Separation of solutes in ion exchange chromatography. The steps for separation in ion exchange chromatography are given as below. 1. The ion exchanger is first treated with alkali, then acid to neutralize it. Finally, it is washed with water. In contrast, the cation exchanger is first treated with acid, neutralized with alkali, and finally washed with water. 2. The ion exchanger is packed into a column and equilibrated with counter ions passing buffer of required pH. 3. When samples containing mixture of compounds is applied onto the top of exchanger bed, the solutes having charges similar to that of counter ions, here negative ions, gene exchanged with the counter ions and binds reversely but strongly to the ion exchange. Figure 3.3a. 4. The other solutes positively charged, and neutral ones, have no affinity for the stationary phase and are washed down along with the buffer B. 5. Thereafter, the bound solutes are released step by changing pH or ionic strength of the elution buffer C. 6. Generally, a buffer with increasing pH and ionic strength is applied with anionic exchanger, whereas with cation exchanger, a buffer with decreasing pH and increasing ionic strength gradient is employed for desorption of bound substances. 7. The ion exchanger is regenerated after passing the buffer continuously. Figure 3.3d. C. Electrophoresis. The charged molecules can be separated by applying an electric field which is called electrophoresis. This technique was pioneered by Atiselius in 1937. When the charged molecules are placed in an electric field, they migrate depending on their net charges, size, shape, and applied current. The velocity of movement of molecules can be presented by the following formula: V is equal to A multiplied by Q. Whereas V is velocity of migration of molecules, A is electric field in volts per centimeter, and Q is net fractional coefficient, which is a function of the mass and shape of molecule. Electrophoresis is applied for separation of RNA, DNA, and proteins. DNA molecules have negative charges. Therefore, based on their size, DNA molecules migrate to anode, i.e., small molecules move faster through the pores of matrix than the larger molecules. Similarly, protein macromolecules are made up of positively and negatively charged amino acids at specific pH. The relative proportion of these two charges govern the net charges of protein macromolecules. If two proteins are of similar size and have identical charges, there shall be a little or no separation of such proteins due to similar charge-mass ratio. Because the electrophoretic mobility of a molecule depends on charge density, charge-mass ratio. On the basis of types of support medium, electrophoresis is of different types such as 1. Paper electrophoresis. A strip of Whatman filter paper or cellulose acetate paper is used as support to separate macromolecules on the basis of their varying sizes. Later on, the spots developed on filter paper are cut and dissolved in solvent for separation and further work to starch gel electrophoresis. Starch is partially hydrolyzed in buffer to prepare a solution. Then it is heated and cooled to get starch gel. The gel acts as molecular sieve to separate molecules. Molecules of varying sizes migrate in gel and separated. 3. Immunoelectrofocusing. It works like agar or agarose gel electrophoresis. It is used for separation of proteins based on charge, mass ratio and their antigonicity. First, proteins are separated, then allowed to react with antigens through diffusion via gel. Therefore, it is called immunoelectrophoresis, agarose gel electrophoresis. Pulsed field gel electrophoresis. Polyacrylamide gel electrophoresis, sodium dodecyl polyacrylamide gel electrophoresis, two-dimensional gel electrophoresis, etc. 1. Agarose gel electrophoresis. Agarose gels are more porous than polyacrylamide gels and have comparatively larger pore size. Therefore, it is used for separation of large-sized macromolecules such as DNA. Agarose is a linear polymer of D-galactose and 3,6-anhydroL-galactose which is extracted from seaweeds. By changing the concentration of agarose, pore size of the gel can be determined. Diagram of an electrophoresis is given in figure 3.4. The negatively charged DNA migrates at neutral pH towards the anode after application of electric field across the gel. It is the standard technique for separation of nucleic acids. Conventional agarose gels cannot resolve fragments larger than 20 kilobits. Because larger DNA fragments are unable to migrate through pores or sieves of gel as small fragments do. 2. Pulsed field gel electrophoresis PFGE. Schwartz and Cantor. 1984 conceived the PAGE to separate the several megabases long DNA molecules. All linear double-stranded DNA molecules, larger than a certain size, migrate through agarose gel almost at similar rate. But agarose gels are unable to resolve linear DNA molecules larger than 750 kilobits in length. PFGE varies a little from agarose gel electrophoresis and can resolve DNA molecules of 100 to 1000 kb. It changes the direction of electric field in such a way that larger DNA fragments align more slowly with the direction of the new field than do the smaller DNA molecules. The pulsed electric fields applied to a gel force the DNA molecules to reorient before continuing to move like snake through the gels. The larger molecules take much time to reorient than the smaller molecules. Figure 3.5. Large DNA molecules are trapped in their tube every time when direction of electric field is changed. Until they are reoriented along the new axis of electric field, they cannot make further progress through the gel. Hence, longer time is required for larger molecules for such alignment. Such DNA molecules are functional according to their size, whose reorientation time is less than the period of electric pulses. Unlike a single constant electric field in conventional electrophoresis, two perpendicularly oriented alternating electric fields are applied in PFGE for separation of molecules. The smaller molecules change the direction quickly, move fastly and separated from the larger molecules. Initially, Schwartz and Cantor 1984 electrophoretically separated the individual yeast chromosomes which ranged in size 230-2000 kilobits. Since then, chromosomal DNA of a wide variety of organisms has been analyzed using PFGE. 3. Polyacrylamide gel electrophoresis. Polyacrylamide gel offers several advantages such as inertness to chemicals, superior resolution, stability over wide range of pH, temperature and ionic strength. Polyacrylamide gel is prepared by using the following: 1. Monomers, e.g. acrylamide, N-methylene bisacrylamide, 2. Initiator, N-N-N-N, tetramethylethylene diamine, TEMED, 3. Propagators, ammonium persulfate or riboflavin, 4. Terminator or inhibitor, oxygen air. The pore size of polyacrylamide gel can be changed by varying the concentration of acrylamide and biacrylamide monomers in a fixed volume of gelatin solution. Free radicals formed by ammonium sulfate activate the acrylamide gel. Long polymer chain is thus produced after reacting the activated acrylamide with successive acrylamide molecules. Further, bisacrylamide brings about gelation and cross-linking through polymerization. This results in formation of complex network of acrylamide. 4. Sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS-PAGE. Some of the proteins are not separated due to similar charge-mass ratio. Therefore, such proteins are treated first with an ionic detergent called sodium dodecyl sulfate SDS before start and during the course of electrophoresis PAGE. Therefore, such electrophoresis is called SDS-PAGE electrophoresis. Identical proteins are denatured by SDS, resulting in their subunits. The polypeptide chains get opened and extended. On the basis of their mass but not the charges, the molecules are separated. Figure 3.6. SDS-PAGE is used universally for analyzing the mixture of proteins according to their respective size. SDS solubilized in soluble proteins makes possible the analysis of the other insoluble mixtures. Separation of the proteins does not occur due to similar charge-mass ratio Z per meter. Therefore, such proteins are treated first with an ionic detergent called SDS before the start and during the course of electrophoresis. Therefore, such electrophoresis is called SDS-PAGE. Identical proteins are denatured by SDS, resulting in their subunits. The polypeptide chains get opened and extended. On the basis of their mass but not the charge, the molecules are separated. Electrophoretic separation is normally used for these reasons. Gel acts as molecular sieves, hence separates the molecules on the basis of their size, and gel suppresses conventional current produced by small temperature gradient, which improves the resolution. Polyacrylamide gel supporting media is used for this purpose due to its good nature, chemically inert, stable over a wide range of pH, temperature, ionic strength, and transparent. Polyacrylamide gel is better for size fraction of proteins molecules. The denatured proteins have a negative charge with a uniform charge to mass ratio Z per meter when treated with SDS, anionic detergent. Proteins migrate toward anode at alkaline pH through PAGE gel during electrophoresis. The smaller polypeptides moves faster, followed by the larger polypeptides. Therefore, the intrinsic charge on proteins is masked in SDS-PAGE. Hence, the separation is based on the size. Mercaptoethanol induces interpolypeptide disulfide bridge and separates the subunits of a polymeric protein. Molecular weight of the separated protein can be analyzed by comparing the molecular weight of the standard protein and its mobility. In analysis of a complex mixture of proteins, the resolution is improved by the initial movement through a stacking gel. The final bands in the separating gel are sharper and focused nicely. 5. Two-dimensional electrophoresis. Protein mixture is separated using two-dimensional electrophoresis. The first dimension uses the isoelectric focusing, and the second dimension is sodium dodecyl sulfate polyacrylamide gel electrophoresis SDS-PAGE. Isoelectric focusing IEF proteins, DNZ and RNA have electric charges which depend on molecule to molecule in the conditions of the medium pH of buffer in which dissolved. Charged molecules can be separated by electrophoresis in gels. Due to the differences in amino acid composition, proteins have a unique mass and charge. Hence, the proteins have net negative charge and net positive charge or isoelectric point, no charge, at a given pH of buffer. The amphoteric substances such as proteins which differ in their isoelectric points can be separated by AF. Isoelectric point is a pH value at which the net charges on molecules are zero. Amphilites, i.e. complex mixture of synthetic polyamino polycarboxylic acids, are introduced into heat gel to create the pH gradient, wide range from 3 to 10, or narrow range of 7 to 8. Then a potential difference is applied across the gel. The molecules having differences in isoelectric points by a little as 0.01 picohenries unit can be separated. Proteins migrate, depending on their charge, until they reach a region which pH corresponds to respective isoelectric point, at which pH proteins possess no net charge and hence got focused. Figure 3.7. B. SDS-PAGE. SDS-PAGE is carried out as described in earlier section. D. Mass spectrometry.